Hi all, welcome back to my channel. Today we'll discuss the histology of cerebellum. There are five main types of neurons present in the cerebellar cortex. We'll first discuss them. The first, the Purkinje cells, which are the flask shaped, present in the middle layer. The dendrite of this will branch like a tree, perpendicular to the plane axis of the folium. Axon arises from the bottom of the cell, pass through the granular layer, and relay in the deep cerebellar nuclei. The next, the stellate cells present in the molecular layer. The dendrites will ramify there. The axon makes synaptic contact with the dendrites of the Purkinje cells. Then the basket cells present in the deeper part of the molecular layer. The dendrites ramify in the molecular layer, whereas the axons runs downwards and anastomose with the pre-axon part of the around 15 to 20 Purkinje cells. Now coming to the granular layer, we have two basic cells, granule cells and Golgi cells. Granule cells are more numerous. The dendrites will branch within the same layer, whereas the axon run towards the surface, and in the molecular layer it will branch like a T shape and run along the long axis of the folium. And it covers a territorial plane of about 500 Purkinje cells. Then the Golgi cells, these are the largest cells in the cerebellum. The dendrites will ramify in the granular and molecular layer. The axon, they make synapse with the granule cells in the rosette. There are two afferent fibers we should know. One is the climbing fibers, another is the mossy fibers. The climbing fibers are the fibers from the olivocerebellar complex, inferior olivary complex. They'll ascend towards the molecular layer and make synapse with all the layer cells there. The mossy fibers consist of all the fibers other than the inferior olivary nuclear complex. So they end in the granular layer. It ends in some 35 to 40 terminal swellings, that is called rosette. This will have a postsynaptic connections with the granular cells and also the Golgi cells. They'll be covered by the cells, Bergman glial cells. Together they form the glomerulus. Now coming to drawing the histology diagram, first we'll make an outline of the folium. You have, as we know, the cerebellum has a lot of fissures and all, so just make an outline of the folium. In the center we'll draw the first white matter, so give some space for the gray matter and then we'll draw the white matter first, so it's easy for us to fill the three layers of the gray matter later. So this is central white matter I'm drawing. First the folium you draw, then the white matter, then shade the background because the connective tissues, glial cells and all will be light pinkish. Don't leave it white. So shade the background. Before drawing in the gray matter, we'll draw the pia matter. Pia matter is the one which closely encloses the cerebellum, the outermost covering in the cerebellum. So it's a very thin connective tissue structure. Some blood vessels also we can show there. You can keep some nuclei to represent the glial cells nuclei, even in the white matter also. Now, coming to the gray matter, usually it has three layers. No, first we'll draw the Purkinje layer where we are drawing the Purkinje cells. Make a light line. Don't make it too dark. So we are going to draw the Purkinje cells on this line. So superficial to that is a molecular layer. Deep to that, between the white matter and this line, will be granular layer. So this is the line where we are drawing Purkinje cells. Don't make it too dark. It's for our reference to draw. Otherwise, the thickness will be haphazard. So the Purkinje cells, make sure it is not triangular. You have to draw like a round bottom, and apical part, the dendrites will ramify. You have to show that ramification of the dendrites. So it's like a teardrop, the water droplets. So round bottom, it's like a flask, narrow neck, and from there the dendrites will branch. So take some time to draw the ganglions, the Purkinje cells. Draw the Purkinje cells first, then it is easy to draw the other two layers. So take some time and draw the Purkinje cells. Don't forget to keep the nuclei in that. So keep the nuclei in the center there. Those are the dendrites ramifying there. So as we're discussing the 5 types of neurons, in that only one type, that's a granule cells, is the only positive neuron. The rest all the neurons are the negative neurons. That you have to remember. Means negative feedback mechanism. When they are stimulated, they inhibit the other neurons. Whereas the granule cells, when it is stimulated, it will positive feedback that will excite the other neurons. So once we keep the Purkinje cells, keep the nuclei also. Now, superficial to the Purkinje cells is the molecular layer, where we have two cells. That is what are the cells? Basket cells and the stellate cells. So superficial part we have a stellate cells, the deeper part we have basket cells. The number is comparatively less compared to the granular and Golgi cells, so keep very few nuclei there. So and also you have dendrites of Purkinje cells, axons of granule cells there. So give some fibers look there, axon and dendrite ramification there. So keep very few nuclei compared to granular layer and some fibers, dendritic connections there with the eosin pencil. So that's a molecular layer. So then we'll finish with the Purkinje cells. Only Purkinje cells form the middle layer. That's a Purkinje cell layer. Now coming to the third layer, that's a granular cell layer. So here the number of granular cells will be more. And also we have Golgi cells, the largest cells. So keep more nuclei there. So that's between Purkinje cells and the white fibers, white matter we have drawn. Keep lot of nuclei. That's a granular layer. So 3 basic layers we have to show in the cerebellum. The molecular layer consists of the stellate cell and basket cells. Purkinje cell layer consists of Purkinje cells. The third layer, the granular layer, consists of 2 Gs. That's one is a granule cells, another is Golgi cells. These are the 3 basic layers of grey matter. Then we have to draw the white matter. So I hope these drawings are going to helpful for you. The cerebellum is the easier slide to identify even without focusing in the microscope. If you see the cross of the slide, you can see the branching pattern, the folium pattern and all, if you without focusing under the microscope. So easy to identify. Don't miss the cerebellum. Draw the diagram properly, even in the exams. So not only granule cells, we have some fibers also ramifying their mossy fibers, climbing fibers. So shade the background pink. I hope the diagrams are helpful for you. If you like the video, give a thumbs up and share the video. This is a label diagram. Thank you for watching.